Welcome back to Bio B160 Principles of Living Systems. I'm Ruth Reitzman, your instructor for the course, and this is part 2 of cell structure. In this episode, we'll continue our tour of cell structure. So let's get started. We'll continue our tour of the cell with a look at 3 important metabolic organelles: the mitochondrion, the chloroplast, and the peroxisome. The mitochondrion is the organelle responsible for cellular respiration. When we breathe, the oxygen that enters our lungs binds to hemoglobin in our red blood cells and is carried to our cells. Each cell consumes the oxygen in cellular respiration, producing ATP, with carbon dioxide and water as byproducts. The mitochondrion is sometimes called the powerhouse of the cell, since it's needed for most of the cell's ATP synthesis. The mitochondrion is made of 2 membranes, an outer membrane and an inner membrane. The inner membrane is highly folded, and these inner folds are called cristae. Remember that folding is a great way to get more surface area, and in this case, there's more membrane to hold the cellular respiration enzymes. This figure shows the structure of the mitochondria with its inner and outer membranes. The space between the 2 membranes is called the intermembrane space, and the area inside the mitochondrion is called the matrix. Later this semester, we'll explore the function of the mitochondrion in depth and learn the details of cellular respiration. The chloroplast is found in plants and algae and is responsible for photosynthesis. The chloroplast consists of 3 membranes: the outer membrane, the inner membrane, and a series of membranous sacs called thylakoids. Photosynthesis consists of 2 parts: the photo part that requires sunlight and the synthesis part that uses the products of the photoreactions to synthesize sugars. All of us rely on the products of photosynthesis to support our life, so be sure to thank the green plants and the algae for their life-giving synthetic activities. An interesting note about the mitochondrion and the chloroplast is their likely origins. If we compare the size and structure of these 2 organelles with bacteria, which are prokaryotic cells, we find many similarities. Both mitochondria and chloroplasts have a double membrane, as do many bacteria. Both have ribosomes and circular DNA, and the size and form of the ribosomes and DNA are similar to the bacterial ribosomes and DNA. Finally, both mitochondria and chloroplasts divide and grow independently within eukaryotic cells. The endosymbiont theory was proposed by Lynn Margulis in 1966 to explain the origins of both mitochondria and chloroplasts. It is thought that the chloroplast was derived from early photosynthetic prokaryotes that were engulfed by other non-photosynthetic cells. The mitochondrion was formed when an oxygen-using prokaryote was engulfed by another cell. In a sense, we have our bacterial ancestors living inside of us. Now let's discuss a third important metabolic organelle, the peroxisome. The peroxisome is an organelle that is specialized for oxidation. Some peroxisomes produce hydrogen peroxide. Others use oxygen to break down fats, which are then further metabolized in the mitochondrion. In plant cells, a specialized peroxisome called the glyoxysome converts fats to sugars. This is particularly important in plant seeds, which live on stored fats as they germinate until they can develop leaves and then use photosynthesis to produce sugars. Our next cellular structures are the cytoskeleton, the structures in the cell that keep it organized and give it strength and shape. Many of the cytoskeleton structures interact with motor proteins to produce motility, both inside and outside of the cell. There are 3 types of cytoskeleton: microtubules, intermediate filaments, and microfilaments. This is a photo taken with a fluorescence microscope and shows microtubules stained with a green dye and microfilaments stained with red. The nucleus, not part of the cytoskeleton, is stained with blue. Note that the intermediate filaments are not shown in this figure, but we'll see them a bit later. Microtubules are hollow tubes made of the protein tubulin. You can see in this diagram how the subunits of the tubulin proteins spiral around to form a microtubule. Microtubules are very important for maintaining the cell's shape, for the motility of cilia and flagella, for chromosome movement during cell division, and for organelle movement. How does a microtubule function? Well, one way is shown here. The microtubule serves as a scaffolding for a motor protein to walk along its length. The motor protein is attached to a vesicle, and as the motor protein moves, the vesicle moves. You can see motility in action if you watch the BioFlix in Chapter 6 resources in MyLabs Plus. Microtubules make up the structure of both cilia and flagella. Flagella are found on protozoa and on human sperm cells. Cilia are also found on protozoa and on the surface of tracheal cells in our windpipe. Another cellular structure that contains microtubules is the centrosome. The centrosome is the region of the cell that serves as an attachment points for the microtubules that attach to and move chromosomes along during cell division. Our second category of cytoskeleton are the microfilaments. Microfilaments are made of the protein actin. Actin subunits wind together to form a microfilament fiber. Microfilaments serve as tension-bearing structures and allow cells to change shape, participate in cytoplasmic streaming, cellular motility, and the cytokinesis stage of cell division. This diagram shows several examples of microfilaments in action. Our skeletal muscle cells have regular arrays of actin microfilament fibers that interact with myosin and together are responsible for our muscle contraction. Microfilaments are responsible for pseudopodia, which is the motility mechanism of the amoeba. And microfilaments attached to chloroplasts inside plant cells are responsible for the movement of the chloroplasts in cytoplasmic streaming. A third type of cytoskeleton is the intermediate filament. There are several types of intermediate filaments, and one type, the keratin filament, is shown here. Intermediate filaments are needed to maintain the shape of the cell and to anchor various organelles in the cell. One type of intermediate filaments is used to anchor the nucleus and to form the nuclear lamina, which lines the interior of the nucleus. Our last topic in this episode is the extracellular components and connections that are used to join cells to form tissues and to coordinate cellular activities. These include the plant cell walls, the extracellular matrix of animal cells, and intercellular junctions. The plant cell wall is made of cellulose and is located outside the plasma membrane. The function of the cell wall is to provide strength and support to the cell. Plant cell walls are joined together through their middle lamella and may form secondary cell walls for additional strength and rigidity, such as those in woody plants. The extracellular matrix, ECM, of an animal cell consists of fibers and proteins that join cells together and also bind proteins to proteins inside the cell. This dense network of collagen and fibronectin proteins are attached via plasma membrane proteins called integrins, which are then tied to the cytoskeletal microfilament proteins. All of this serves to keep our tissues tied together. Another feature of tissues are the intercellular junctions shown here. Tight junctions, desmosomes, and gap junctions help seal cells together and allow for communication between cells. Now that we've looked at the structure of the cell, we can talk about how these structures coordinate together to accomplish a particular function. In the photo here, a macrophage, a type of white blood cell, is engulfing bacteria, which will then be destroyed by phagocytosis. Can you list the organelles needed for a macrophage to engulf and digest a bacterium? Something to think about as you prepare for the first exam. This chart on page 123 summarizes the structures found in eukaryotic cells along with their functions. This will be a very helpful diagram in reviewing this chapter's materials. In this episode, we've explored cell structure and function. You should be able to distinguish between prokaryotic and eukaryotic cells. You should be able to identify the structures shown in figure 6.8 and describe the functions. You should be able to describe the structure and function of the endomembrane system components. You should be able to briefly explain the roles of the mitochondria, chloroplasts, and peroxisomes. You should be able to describe the three types of cytoskeleton and describe the extracellular connections of animals and plant cells. This concludes part 2 of cell structure. To continue your study, be sure to do the homework on the MyLabs Plus Mastering Biology site and post your questions to this week's discussion board. Also, be sure to watch the BioFlix on the tour of the animal and plant cells in the Chapter 6 resources in MyLabs Plus. Thanks, and have a great day.